So yeah, our next speaker is Rebecca Akarasola, a final year PhD student in statistics at the University of Edinburgh. And I'll let you get started, Rebecca, and you can tell us what you're going to be talking about.
Uh, Okay, thank you very much. Thank you for inviting me over for the talk. So I'm going to share my screen quickly and let me know if we can see my screen. Can we see my screen?
Yeah, we can.
Okay, yeah, so thank you very much once again. So I'm Rebecca, a final year PhD student at the University of Edinburgh. So I'm going to be giving a talk based on part of my research. So for this webinar, I've titled it Observing the Unobservable. Okay, so I give us a quick background to this part of my research. So it focuses on seabirds, where we're trying to infer the behaviors of seabirds from their movement tracking data. And unlike the previous talk where it's from the perspective of the Bayesian framework. Here we're looking at the frequentist framework. Yeah, so seabirds, their population are experiencing decline faster than any other kind of bird due to so many reasons, some of which are, based on human impact, such as we have, overfishing, we have construction of offshore wind farm, we have, bycatch, so some of these reasons have led to the decline of their population. And where my research comes in is like we're trying to understand how we can, preserve this population by knowing where these seabeds forage such that these areas can be protected against human impact. Looking at the video on the left hand panel, we can see an example of a movement track across the globe. And so the study of movement ecology, where we look at the study of movement animals, so we're interested in where an animal has been to, the time the animal was at a given location, and most importantly, why the animal is at that given location. Is it there to feed, or is it resting, or is it swimming, or depending on what the animal is doing? So looking at this video on the left-hand panel, we can easily know where an animal has been to, but you'd agree with me that we cannot directly answer the question of what the animal is doing by just looking at the movement tracks. So in the context of the marine environment where my research focuses on seabirds, it's quite difficult to to directly answer the question of what the seabirds are doing by looking at their movement tracks. So in practice, these behaviors are usually inferred from the movement tracks of seabirds retrieved from GPS devices attached to these animals, and these inferred behaviors are rarely validated. Also, we can infer these behaviors from their movement tracks because the way an animal would move when it wants to feed would definitely be different from the way the animal would move when it's just going to a particular location. So due to the difficulty of accessing the first-hand behavioral information of animals, and in the context of my research, we're looking at seabirds due to the terrain of the marine environment, the inferred behaviors are usually difficult to validate, and this is because we cannot, easily access this behavioral data to be used for validation. But so the goal of this part of my research is to, validate the inferred behaviors of seabirds for effective conservation management. So how do we go about this? So we had access to a very unique kind of data set from JNCC, that's the Joint Nature Conservation Committee, where they used a method called visual tracking to, get the information about, the behaviors of the seabeds that are being tracked, and also, the locations of the seabeds. So how is this done? Using the image on the right-hand panel, so I'll be using this to just give us an insight into how this visual tracking was conducted. So it was conducted within this year, 2009 to 2011, where we have like some set of observers on the boats, trying to, mimic or replicate the movement of the terns that have been visually tracked. So once the sites return, leaving the colony, the observers on the boats, begin their tracking of this particular of an individual tern per time. So they try to replicate the movement of this tern such that, the movement of the boats. So the person operating the rib is trained such that, it replicates or mimics the movement of the tern, and the observers on the boat also notes down the change of the behavior of the terns during the visual tracking trip. So because of this kind of method used in collecting this information, we have access to the observed behavioral data of these seabirds that are visually tracked. And also, we have the location data of the boats because of the onboard GPS. And we use this location of the boats as a proxy for the location of the terns. And for context sake, the sampling interval was 1 second. Now, the visual tracking method that I explained was conducted across these different breeding colonies in the UK during the chick rearing season. And for my research, these are the species of seabirds that were visually tracked. So we have 10 species, so we have 4 of them that were visually tracked across all of these breeding colonies that we have here in the UK. So how is the workflow of my research for this particular explanation that I've given? So firstly, we're going to be looking at assessing the visual tracking data. This is because, in practice, behaviors are usually inferred from, the movement tracks of animals gotten from attaching GPS devices to these animals to record their locations. However, in our case, we are using like a proxy location for these terns, which is the actual GPS location of the boat. So it is important that we assess that this actual location of the boat is a good proxy to the location of the terns, given that a reasonable distance was left between the boats and the terns that have been visually tracked. So we go on to, fitting hidden Markov model, which is one of an appropriate statistical tool that can be used to infer behaviors from telemetry data. Then after we fit HMMs, we infer behaviors and we then validate these inferred behaviors using the observed behavioral data that was gotten from the visual tracking trip. So we go straight into the first step, which is, assessing the visual tracking data. So how do we go about this? So like I showed earlier on, this visual tracking trip was conducted across different breeding colonies in the UK during the chick rearing season for different species of terns. So, but for a particular colony, we had access to some information. So with that information, we wanted to check how well the actual position of the boat can be a good proxy to the actual location of the terns that have been visually tracked. So how do we go about this? So some of the information that we had access to for that particular colony, we had access to the distance between the boats that was used for the visual tracking and the particular individual tern that was tracked at that particular time. So we had information about the actual GPS location of the boat and also the bearing between the boats used for the visual tracking and the individual tern that was tracked at that particular time. So with this information, we were able to get an approximate locations of the tern that was being tracked at that particular colony. So here's just an example of some of the results that we looked at for assessing the boat location data. So we compared, the movement tracks of the boats used for the visual tracking to the approximate tern track that we computed. And we observed similarities for all of the individual terns that were tracked for that particular colony, that we had access to the information used to compute the approximate tern location. So we also went ahead to look at how similar the inferred behaviors are for these two categories of data set, which is the boat location data and the approximate tern location data that we were able to compute using the information that we had. And we saw similarities in terms of the behaviors inferred from these two categories of data set. So now having checked the box that okay, the boat location data set that we are using is actually a good proxy to the actual location of the terns. So we then went ahead to the next phase of the work, which is fitting an appropriate model to the visual tracking data in this case, which is the GPS location data of the boat, to infer behaviors of the terns from our fitted models. So I'm going to be giving a very, very, very basic introduction to hidden Markov models, but at the end of my talk, there's a link that we can get access to a full detailed explanation of hidden Markov models. So what is a hidden Markov model? Like I showed in the video earlier, we have the movement tracks of animals that are influenced by the behaviors of the animals, the way an animal would move when it wants to feed. For instance, we have seabirds, the way it's going to move when it wants to forage or catch a fish from the sea would definitely be different in terms of the pattern when it is going or returning back to the colony. So When we attach GPS devices to these animals, in our case, seabirds, we retrieve the movement tracks of these animals. And when we plot our movement tracks, we will see some patterns, which are, these patterns are influenced by the behaviors of these animals that we are trying to infer. So we can say that a very, very basic definition of HMMs, we can say HMMs, is a time series model that consists of two processes where we have the state or the eating process. Now, in the context of our research, the state or the eating process in our case is the animal behavior, which is the behavior of the seabirds that we cannot directly observe given the terrain of the marine environment, while the observed or the state dependent process in the context of my research are simply, the animal's location, or in our case now, the location of the boat that was used in visually tracking the individual turns. So going to the relationship between these two processes that I have explained. So what is the relationship between the state process and the observed process? Firstly, for the state process, we have, that's the probability of being at a particular state at a particular time only depends on the immediate previous state and not on every other state. Using this image to explain what the mathematical expression that we have here, let's say, for instance, this is time t. The state of this current turn at this particular time depends only on the state at the immediate previous time, that is, let's say, t minus 1, and not on every other previous state, that is not on these other two previous states that we have here. And for the next relationship that we have, so what is the relationship between the observed process and the state process? The relationship between these two processes is simply that, the distribution, of being at a given location at a particular time only depends on, the current state at that particular time and not on any other state. So that's like the basic explanation to show the relationship involved between these two processes for hidden Markov models. So how do we introduce this framework in analysis? So for the context of our research, we used the popular R package used for fitting HMMs to animal movement data, which is the momentum package. And how do we introduce the observed process that I explained earlier into the HMM framework? So for the observed process, I mentioned that in our context, the observed process is simply the movement tracks of the boat that was used in visually tracking the tent. Well, however, in practice, it can be, the movement tracks of the animals that we want to infer their behaviors. So when we have access to the movement data, we can, convert this movement data, which is, either longitude or latitude, to, to appropriate movement metrics that can be used in our analysis. And in our case, metrics that we're considering is the metrics of the step length and the turning angles of the boat that was used in visually tracking the turns. And in introducing this into the Eden Markov model framework, we specify distributions for the step length, and in practice, we usually use the gamma distributions for the step length at the different states that we want to infer, while for the turning angles, we can use the von Mises distribution. So how do we introduce the state process into the HMM framework? So for the state process, like I explained earlier, in our context, the state process is simply, the behaviors of these seabirds that we want to infer from the movement data. And in our research, we're focusing on just two states, which is, the foraging state and the traveling state, or the not foraging state. So we simply have like a transition probability matrix is what we use to introduce the state process into the HMM framework. So if we have two states, definitely we have a 2 by 2 transition probability matrix. So if we have n states, we simply have n by n transition probability matrix. So I haven't explained the basic introduction to HMM. So for our research, we looked at two-state HMM. Like I mentioned earlier on, fitted with the popular package Momentum for fitting HMMs to movement data. And we looked at 6 different models with model 0 as our baseline, where we looked at the effect of the pooling effect and the covariate effect on the state process and the observed process. And this table here, just explains or summarizes the different varieties of HMMs that we fitted to the boat tracking data, which is our observed process in this case. And for the covariate that we used is simply the distance of the boat to the colony, while in practice it can be the distance of the animal to the colony. So here's just an example of one of the results from, HMMs fitted to the observed process, which is the movement tracks of the boat. We can see on the left-hand panel, we have the distribution of the step length fitted by the HMM fitted state-dependent distribution. And as you can see, for the state-dependent distribution, which is the foraging behavioral state in orange, we can see it's characterized by fewer step length, while for the not foraging state or traveling state, we can see that it is characterized by larger step length compared to the foraging behavioral state. So for the turning angle, as you would see that for the foraging behavioral state, for the traveling behavioral state, which is indicated with blue, it is more centered around zero, because when, and let's say for instance in our context, when the seabird wants to feed, for instance, catch a fish in the sea, would have fewer step lengths and more turnings compared to when it is, let's say, going back to the colony, would have larger step length and lesser persistent in turning. And here's just an example again from the varieties of models that we fitted to 4 different species of tens across the different breeding colonies that we have in the UK. So here's an example again showing, comparing the movement tracks, colored by the on the left-hand panel, we have the observed state, which was not used in our analysis, and the observed state was gotten, by the observers on the boat when they were visually tracking the turns. While on the right-hand panel here, we have, the movement of the boat, decoded or colored by the inferred, behavioral states from our HMMs, and we can see similarities to the observed behavioral state. So now that we've fitted HMMs to the visual tracking data that we have, and we have also inferred our behaviors from this fitted model, so we now want to validate these inferred behaviors using the observed behavioral data that we have access to due to the peculiarity of the data provided by GNCC. So how do we go about validating these inferred behaviors? Because like I mentioned in practice, behaviors inferred from from HMMs are rarely validated, and this is because we usually do not have access to the true behavioral information of these animals. And in the context of my research, we're looking at seabirds in the marine environment. And if we can't really validate these inferred behaviors, it may impact conservation management. So how do we go about validating these inferred behaviors, given that we have access to the true behavioral information of these turns that were visually tracked? So it's very straightforward. Given our observed behavioral information that we have and the inferred behavioral information that we retrieved from the HMMs, we simply use the metrics of the F1 score to validate our results. And here is just summarizing the validation results that we have for all of the breeding colonies and for the 4 different species of tens that we have. And for the result indicated with the green is just simply the overall performance of the HMMs in decoding the behavioral states, where we're looking at 2 states of foraging and not foraging. And we can see a fairly good performance across all of the species of tens and from all of the colonies. While for the result indicated in orange, we're focusing on just the foraging behavioral states, the performance of the HMMs in decoding the foraging behavioral state, because in terms of conservation purposes, this is usually more of interest, because when we have an idea of where these seabirds or where these tens forage, these areas can be mapped out and can be protected from from negative human impact. So this is our validation result that we have. So to wrap up, from what we've looked at so far, like I mentioned in practice, these inferred behaviors are rarely validated due to the difficulty in accessing, the true behavioral information of these animals. And HMM is a common, statistical method that are usually, applied to telemetry data to infer these behaviors from telemetry data. But given the work that we've done, there's an increased confidence in, unvalidated inferred behaviors that are usually, gotten from HMMs fitted to telemetry data. And also, it also increases our confidence in the sense that, these inferred behaviors from HMMs can help, to inform, spatial planning and also designing of, protected areas, just to limit the impact of negative impact of humans. So that's all for what we've looked at for a particular aspect of my research. So if you're interested in knowing more about, learning more about the research, particularly learning more about HMMs in a very high level definition, we can look at these references. And also, if you want to learn about using the package for fitting HMMs to telemetry data, we can also look at these references as well. So thanks to my supervisory team and to the funding body, and also to GNCC for providing the data, and also to the Young ISA for inviting me for this talk. So thank you very much for your attention. Questions are welcomed. And if you are interested in learning more about the research, we can scan this QR code, which will grant you access to our published paper. Thank you very much.
Thanks, Rebecca. It's a great talk, really interesting.
Thank you.
Do we have any questions? I have a question, but I'll
Okay.
see if anyone else has questions. Do you want to go first?
Me? Okay.
Sorry.
Yeah. So short question. Or
Okay.
couple of short questions, I guess. Um,
Okay.
the HMM process,
Mhm.
so before that, are the data coming in at regular intervals?
Yes, the data are coming at regular intervals because that is one of the criterion that must be satisfied for us to be able to make use of HMMs.
Yeah.
Yes.
Okay, so you're using essentially a discrete time HMM.
Yes. Yes.
What's the frequency of the data you're getting? Is it like
Okay,
every few
so
seconds or every few minutes?
every 1 second.
Okay, so you could, in theory, just model it as a continuous time Markov process, a continuous time Markov chain. Right?
Yes.
Yeah, I'm just trying to think like how if it would be much different. Do you think it would be much different in terms of like the model flexibility, or would just kind of give you the pretty much the same results?
We should get the same result, hopefully. Yeah, but
Yeah.
we've not looked into, fitting with continuous HMMs, time HMM.
Okay. Yeah. Thanks.
Okay.
My question is much more specific. That
Yeah.
you showed two graphs. One was the true data and one was the HMM prediction. And there's one part of it where the HMM predicts that they're foraging, but the true data shows that they're not. And I'm just
Okay.
interested to know, um, do you have any insight into maybe why that was the case?
Is it this particular slide?
Um, no, it's further on. You actually had graphs of the movement. 20, I think. Slide.
Okay.
See that like kind of longer section where they go much further out and it's kind of around the back of this island. You've got like three
Yes, yes,
of
yes.
foraging. I think that's just very interesting because they're kind of, that's a point where they've seemingly traveled quite far
Yes.
and
Uh,
then obviously just turned around and came back. No interest
yes.
in period anymore.
Yes, so I think the reason for this may be that, so for this particular data set that we're working with, it's the, GPS location of the boat that was used in tracking the turns. So one of the limitation is that, the way a turn will sharply, turn, probably to catch a fish from the sea, may not be directly and easily replicable by the operator of the boat used in tracking the turn. So given this observed behavioral states recorded by the observers on the boat, so they may not really catch, that very, very quick and swift change in the behavior of the turns that have been visually tracked. And on the right-hand panel, the HMMs would actually, pick that up given the nature of the parameters that would specify. So probably that might be a reason why that is the case, due to human error or due to the fact that, the operator of the rig may not directly replicate that sharp turns or movement of the seabeds that have been visually tracked, or the observers on the boat may not, note down that particular very swift change and the behavior of the turns that have been visually tracked. So that's maybe one of the reasons why we have this issue.
No, it's just interesting. I thought it
Yeah,
was strange that they went so far and did nothing.
yeah.
Do we have any other questions? I think we probably have time for one more.
Okay.
Otherwise, we can say thank you, Rebecca, and we'll
Okay, thank
move
you
to
very
the
much.
discussion.
Thank you.